Hello friends, welcome to STEM biology class. Friends, today we shall discuss about a primary active transport system known as calcium ATPase. Calcium control critical cellular responses in all eukaryotic organisms. It controls both short-term biological processes that take milliseconds such as muscle contraction and long-term processes like cell proliferation and organ development. The plasma membrane contains several types of channels that mediate the entry of calcium ion into the cell. The plasma membrane also contains two systems that mediate the removal of calcium ion from the cell. These two systems, which are responsible for the removal of calcium ion from the cell, are a sodium calcium exchanger, which is known as NCX, and another system is plasma membrane calcium pump, which is also known as PMCA or plasma membrane calcium ATPase. In addition to this, a form of calcium ATPase is present in the membrane of sarcoendoplasmic reticulum, which is known as SERCA pump. The plasma membrane calcium ATPase, known as PMCA, has been attributed a housekeeping role in maintaining the cytosolic calcium ion. It is a P-type ATPase. It is considered as the fine tuner of cytosolic calcium ion as it operates in a concentration range in which NCX is relatively very inefficient. NCX has been attributed a dynamic role of counteracting a large variation in the cytosolic calcium ion in the excitable cell. NCX and PMCA mediate the removal of calcium ions from the cell, but the calcium is also restored to its basal level by sequestration in the sarcoendoplasmic reticulum via SERCA pump. SERCA pump and plasma membrane calcium pump have the same general features. Both of them have 10 transmembrane domains and a large cytosolic headpiece that is divided into three important cytosolic A, N and P domains. The plasma membrane calcium ATPase was discovered in erythrocytes by Salzman in 1966. It was then described and characterized in numerous other cell types. Structurally, the PMCA is predicted to consist of 10 transmembrane domains, 2 large intracellular loops and 1 N-terminal cytoplasmic tail and another C-terminal cytoplasmic tail. Friends, this figure is the topology model of plasma membrane calcium pump. This is the plasma membrane and these are the 10 transmembrane domains. Between transmembrane domain 2 and 3, there is a large intracellular loop. Similarly, between domain 4 and 5, there is another intracellular loop. This is the N-terminal cytoplasmic tail and that is the carboxyl terminal cytoplasmic tail. The N-terminal cytoplasmic tail is about 90 amino acids long. It has a consensus binding site for 14 3,3 proteins that play an inhibitory role on pump activity. This is the intracellular loop which is present between transmembrane domain 2 and 3. This loop is considered as the transducer domain. This loop contains one of the two sites that mediate activation by acidic phospholipid. It also has one of the two sites for autoinhibitory interaction with the calmodulin binding domain. This intracellular loop also has one of the two main sites of alternative splicing that generate pump variants. PL is the acidic phospholipid binding domain. The second intracellular loop connecting the transmembrane domain 4 and 5 contains the catalytic center that includes a ATP binding site and a aspartate residue that is phosphorylated during the course of action. This intracellular loop also contains a second binding site for carboxyl terminal calmodulin binding domain. The other important component of the plasma membrane calcium pump is the carboxyl terminal intracellular tail, which contains the main regulatory sites that include a calmodulin binding domain, consensus sites for protein kinase A and C. It also has a high affinity allosteric calcium bind site. The calmodulin binding domain, which is represented by a rectangle, acts as an autoinhibitory domain binding to specific sites in the first and second intracellular loop. The carboxyl terminal region of the plasma membrane calcium pump is also the target for isoform specific phosphorylation by protein kinase A and C. It also contains an important site for alternative splicing known as site C. The calcium ATPase actively pumps 2 calcium ions out of the cytosol at the expense of ATP while counter-transporting 2 or 3 protons. This figure explains the mechanism of pumping cycle by the calcium ATPase, which is present in the membrane of sarcoplasmic reticulum. The mechanism of action of calcium ATPase closely resembles that of Na plus K plus ATPase. This transport system exists in two conformational states known as E1 and E2. 2 calcium ions bind within a bundles of 10 transmembrane domains. The structural differences between E1 and E2 indicate that both the intracellular domain and the transmembrane domains undergo extensive rearrangement during the transport cycle. Friends, this is all about calcium ATPase. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you.